talk today is about what we've done as a company to address what is coming our way in the future. So I'm going to show you the first slide. First of all, I want to show you that little logo in the bottom of right corner. Yeah, this year is the 40th anniversary of Biogen. So it started 40 years ago. So Biogen, this is not to advertise what we do, is to tell you why we are doing the innovation that we are doing. So our mission is very clear. We are pioneers in neuroscience. Now, when you think about neuroscience, you think about many things. So keep these concepts in mind. You think about the CNS. You think about how you're going to reach that. You think about how much product you need to actually get to the brain. But why are we doing this? If you look at all of the neuroscience or neurodegenerative disorders that are there today, there's 1 billion people that are affected by neurological disorders. So you have dementia, like Alzheimer's, you have Parkinson's disease, multiple sclerosis, and so on. So it's a huge problem, and we believe neuroscience is today where oncology was many years ago. There's a lot that we don't know about the science and the drugs that we need to get to work in this space. But we know that we need to get to the CNS. So it has a huge impact to the healthcare system. And as you know, and I have my own, all of us have been touched by dementia in our either parents or grandparents. It's a huge problem for our society, not only for the people that are suffering from dementia, but also for the caretakers and the healthcare system in general. So what we've done as a company, we develop a rich pipeline of products that allow us to treat these disorders. But what the pipeline has multiple modalities, and actually we have focused on 4 particular modalities in our neuroscience space. So we have small molecules, biologics, antisense oligonucleotides, and gene therapy. And as a company, we are modality agnostic. So we will look at the modality, the kind of candidate that will allow us to treat the disease, the particular disease that we are interested in. And for some of them, like dementia, we're actually using 3 of those 4 modalities. So we have proteins or biologics, we have antisense oligonucleotides, we're also looking at small molecules in the dementia space. And in general, we will look at the modality that is best suited for the particular disease. But today I'm going to talk about biologics or protein therapeutics and what we are doing in that space. And an important concept here that has been covered by many speakers today and by my speaker before me is efficiency and agility and speed. It's very important to get us to where we need to be so we can get that to go from molecule to patient. So what have we done at Biogen from a manufacturing point of view to address our future and what we need to produce? So not only are we modality agnostic, we are also a manufacturing platform agnostic. So everybody says Biogen is building this big plant in Switzerland with a lot of stainless steel. I'll tell you why we're doing that. But we also have flexible volume manufacturing, so we have a fully disposable facility in an RTP campus that we use for clinical manufacturing. We also operate at a 2,000-liter scale, and we also operate at a 15,000-liter scale. And we do a lot of modeling to determine what is the right scale for the products and the processes that we have. So gene therapy is all disposable manufacturing. For some of the proteins that we need to reach the CNS, we actually have to use stainless steel technology. And you can see some of the kind of analysis that we do on the graph in there, where you can see, if we have to make today metric tons, and actually we need to make more in the order of 10 or 20 metric tons, so that's 20,000 kilos of monoclonal antibody a year. You can't reach that with a fully disposable facility like we have. So to lower the cost of goods, that is one of the topics, the top 5 that we heard at the beginning, right? We actually have to use stainless steel technology. So depending on how much we need to make and of what modality is what technology we will use. So in our manufacturing network, we have 3 sites. We have our sites in RTP. There we have all 3 technologies. So we have fully disposable, we have 2K, we have 15K. We also do drug product manufacturing there. And we also do antisense oligonucleotide manufacturing in our RTP campus. In Hillerød in Denmark, it's a drug substance, biologics drug substance facility, high throughput production, commercial products. I should have mentioned in our RTP facility, we do clinical and commercial manufacturing, and it's a launch site for most of our products. And then we are building a facility in Switzerland. The town is called Lutherbach. The canton is Solothurn, so you hear both names that we use sometimes. And what we've done in that facility, we have put all of our technology innovation over the past many years. And what I want to do in the little time that I have left is tell you why we did that and how we did that. So when we started looking into what we needed to, how much we needed to produce for our pipeline products, especially in the Alzheimer's space, we did modeling. And here you can see in this modeling, if we look at the current capacity, we could not meet the demand that we will have with existing capacity. So we knew we needed to build additional capacity beyond our campus in North Carolina and campus in Denmark. And we knew that our pipeline had even more products like that. So we started modeling with time and said, we are going to have to build. And we're going to have to build fast, and we're going to have to build large if we really are going to meet that demand. And so, but if we stay with the technologies of today, for that demand, instead of having 2 facilities, we'll probably have to build 10 facilities. And you know how much these big stainless steel facilities cost. I don't need to tell you that. And so what we did is that we got together and said, what are all the innovations and all the technologies that we have developed as a company in our process development organization and our innovation groups? And which one of those we can put together so we can actually build a facility that is not 10, but is actually 1, and allow us to meet that market demand. So that's what we need. And we put all our innovation into what we call our next generation manufacturing. So next generation manufacturing for each company means something different, because the needs of the company are different. Our need is to make a lot of protein, right? We were talking about 10, 20 metric tons of protein that we need to produce a year. So how are we going to make that? So that's what we call next generation manufacturing. And the challenge that we were given is to deliver meaningful therapies for 1 million patients with Alzheimer's disease by 2020. At 1/5 the cost of traditional biologics manufacturing. So not only scale up in how much we have to make, but significantly lower the cost of what we are manufacturing. And so we use a lot of modeling. You heard about modeling in the previous presentation. So we use a lot of modeling to figure out what is it that we need to do. And then we looked at all the innovations, as I mentioned to you. So what we did on the process side, we implemented what we call our 3X platform. 3X means we increase our output by 3 times, right? That's what 3X means. So we worked on increasing the productivity of our cell lines. We also implemented N-1 perfusion technology, so we can seat the main reactor with more cells. We do high-capacity purification. We use buffer concentrates. We also are doing concentration with single-pass tangential flow filtration. When you put all of these technologies together, that were part of what we were doing in one product or another product, but we combined them all in one, in our platform of the future, we were able to increase our output by 3 times. Key to that, and you may have seen presentations in this conference and others, was the increased productivity of our cell lines. We couldn't get to where we are today if we had not done that work. But combined with all the others, process and equipment technologies that I mentioned, we were able to get to that 3 times outputs. The other thing that we did is we look at process control. And again, the previous speaker talked about variability. So one of the things that we wanted to do is minimize variability. So in our legacy processes that we have, if you have variability in your input, you are bound to have variability in your output, and even larger variability that can get you into an out-of-specification situation, or in your product testing, or just being outside of your parameters to operate your process. So what we set out to do is what we call our Advanced Process Control Initiative. How can we control the process so we can not only minimize the input variability, but also minimize further the output variability, and really control our process very tightly. So what are the elements of that? So again, our intent was to have a well-understood and controlled process that will drive robustness and consistency, quality, improve efficiency, and also give us real-time quality. So the aspects of our advanced process control strategy were on raw materials control, making sure that we understand our raw materials and understand the availability of the raw materials. We work with our suppliers. We develop tools to screen raw materials. We work with our suppliers to give us the certificate of analysis of our raw materials, where some of the tests that we develop are actually done by our suppliers. So we really understand, we look at genealogy of the suppliers to our suppliers, and some of that was described in the previous presentation. We also looked at using more PAT or PAT technology, not to monitor that we have done in the past, but to actually control our manufacturing process. And I'll show it to you in a couple of slides later. We also wanted to do what we call an adaptive process control. Can we look at our process and understand what is happening with the process and be able to correct any variability that may be there through an adaptive strategy? We also wanted, and in a previous presentation, I was in the cell therapy session, and people talked about our real-time release testing. And what we said to ourselves, so for that you need to do inline, at-line, online tools, right? Not everything is inline and online. And sometimes you need at-line tools. And what we renamed this is right-time release testing. So do I need to have that number in seconds, or can I have it in an hour or 2 hours? And that's a kind of right-time release testing that allows you to do use at-line tools to do product testing. And then we move the testing to the critical control point instead of accumulating it in the drug substance release testing that is a traditional way of doing testing for drug substances. And lastly, we use models. And I think the previous presentation covered a lot of how you do predictive modeling. But we can predict product quality based on parameters and a lot of the data that we collect from our manufacturing process. How do we leverage predictive modeling to make sure that we can control and adjust the process if necessary? So those were all the principles in our advanced process control strategy. This is a little bit of our raw materials that I described earlier. And so we have different levels of what we are doing in our approach to our raw materials strategy. One of the things that we wanted to do is make sure that the incoming raw material could be tested in a day versus the long time that we all have when the raw materials arrive in our facilities. And for that, besides asking our suppliers to run some of the assays that we want to use to make sure that raw materials meet the attributes that we want for our manufacturing process. So what we do is rapid ID testing in our manufacturing plant. So we either use Raman or X-ray fluorescence to that rapid ID testing, and that takes us about a day. And so what are all the innovations that within the APC process? So for the upstream innovations, as I mentioned to you, besides we significantly enhance the way we screen for clones and develop our cell lines, but we also did the N-1 perfusion to allow us to inoculate the big reactor with more cells and achieve a much higher cell density and much higher output productivity. We use a capacitance probe not to monitor the cell viability in the bioreactor and the viable cell count, but to actually control the feeds. So instead of feeding by day, that is the traditional legacy processes, we feed by need. We measure how many cells we have, and then we feed with an algorithm accordingly how much nutrient. And we also separated the glucose feed from the nutrient feed and control the levels of glucose to a very low level with a Raman probe, and also do the glucose feed based on the reading of the Raman probe for glucose concentration. And we also do raw materials control. And the hypothesis here is that we can tightly control the growth, and we have repeated growth behavior, right? Because we are controlling how we feed. So we don't underfeed, we don't overfeed. That is usually what gives you variability in a process, because we are feeding by need. And the growth curve is repeatable because we control the raw materials, we control the feeds. Then we will be able to get consistent product quality. And to make sure that we get the consistent product quality, that's what we use our predictive modeling to. On the downstream, I think I mentioned some of the technologies that we implemented, and they are described in here, but we are using high-capacity resins for like the capture column. We use flow-through for the other columns, and some other innovations that you can see in this slide. I'm rushing a little because I have a clock reminding me how much time I have left. One of the things that we did is look at feed-forward control. So when we get to these real high titers, so our manufacturing process in the fed batch mode is giving us about 11 grams per liter, 12 grams per liter. When you get to those really high titers, you're also having a lot of protein in your bioreactor. And one of the things that we need to make sure that we control is the level of aggregates, because also we have high concentration protein formulation. So we are going to the 150 to 100 mg per ml in the drug substance. So now you have a process that in the bioreactor has a lot of protein, and then you have a last step that concentrates significantly. The level of aggregate and controlling that is very important. So one of the things that we've done, this is one of our molecules, is to measure quickly, determine the level of aggregates before the last column, and feed forward control and adapt the number of cycles that we run in the column. That means the load of what we're going to load that column with, depending on the level of aggregate. So you can see there, if it's below a particular threshold, we may run 3 cycles. If it's above, we change the number of cycles that we run in that column. So that's feed-forward control. And the other thing that we do is predictive modeling of product quality, so we should be able and are able to predict that aggregate level. We measure it, but we can also predict it. And just like the previous speaker mentioned, FDA has been very welcoming of our predictive modeling capabilities and how we can use it. Other agencies are a work in progress, I would say. But here is an example of one of the models for one of our product quality attribute, the actual data points, and then the predictive model in the graph with the green and the black dots. So you can see how those look like. So this is our process that we run. I described most of it to you, but now it's put all together. So we use feedback control in the bioreactor for the feeds, so that we feed by need. We do feed-forward control for the level of aggregates. We also control the UF step and the concentration step by inline probe to measure protein concentration. But the other thing that we do with data, and I'm glad that I don't have to talk a lot about that because you heard a lot in the previous presentation. We acquire a lot of data too, and we use Simca Online for all our manufacturing processes to monitoring the processes real time. But we also use that same multivariate analysis not only to monitor on real time the process, but to also look at consistency. And you can see a consistency, a principal component analysis consistency plot in there. So we can look at each per unit operation. We can also look at the full manufacturing process. Because the other thing that we've done in the manufacturing is that we have included, we have a fully automated process. And with that, to make sure that, so we review by exception, right, our production. And so another assurance that the process ran properly is to look at consistency plots that you can see in there. And how does a certificate of analysis will look like? So we do a lot of in-process testing, right? So instead of having a QC lab that does a lot of drug substance testing, we do either in-line, at-line, and we only have a high-tech lab or what we call the high-tech lab for purities, and so the peptide map, any kind of binding assay for biological activity, and the safety assays. All the other assays have moved to the critical control point, and they have moved to, and I'll show you our manufacturing facility. They've moved to what we call the Witt Labs, right? The work in progress labs that we have throughout the facility. So all of these things we put together in our facility that we're building in Switzerland. But the other thing that we did, we said we are going to get somewhere with process intensification. That is what I described to you until now. But we also need to improve our productivity through operational excellence. So that's a big component too of improving productivity, and that's why a manufacturing organization comes into play. So process optimization, automation, and I'll describe some of those. Lean operating systems, systems, and so on. And so what is a new way of working? I talked about the manufacturing technologies. I talked about the advanced process control, but we also have what we call high-performance operations. So the testing that I told you, we localize, having technology at our fingertips. So we have in each manufacturing area, we have what we call a collaboration room where all the data is accessible to the operators and also the engineers that are working in the facility. And we make decisions. So our goal is that no sample rests, and all the decisions are made on the floor, real time, as the data is available to us. And you can see it in here. And I think I mentioned this one too. So let me show you a little bit of how a facility looks like in the few minutes that I have left. So we have, as I said, we've increased our level of automation, and we have electronic batch record systems. You can see how an operator will be looking at the Delta V screen and MES and the right hand side. And the other thing that we've done is we've integrated all of our electronic systems, and we have a recipe-driven operation. So we have one recipe per product that drives all of these different systems that you're all familiar with, because we all have them in our facility. So Oracle is our ERP, but we also have Swisslog for our warehouse, and you can see the Delta V Syncade for our MES system. You have the electronic, you have Labware for LIMS and the ELN, etc. You can see Simca Online in there, Statistica, Pi for our data historian. All those systems are integrated in our facility to run from a recipe. And with that, we are able to achieve that lowering the cost. So the cost lowering comes from productivity increases, but also comes from manufacturing optimization for how we operate. And we, by review by exception, by not having batch record review, you need less people, right? You're not doing manual batch record review. This is all review by exception. So you start adding up, and that's how we get to that 1/5 of lowering the cost of our production by 1/5. So let me show you briefly the plant. It's in Switzerland. We have what we call a biomanufacturing cell concept. So we have 4 18K bioreactors, 2 seed trains, and 1 purification in each of the biomanufacturing cells. We did the modeling to determine what was the appropriate ratio of bioreactors to purification, and we settled on 4, and 4 bioreactors to 1 purification. Each of these cells makes 10 metric tons of monoclonal antibody. So they are designed to take up to 15 grams per liter of product out of the bioreactor. It's optimized for that 10 plus gram per liter, but we can also go all the way down to 2. So we have a lot because we know our pipeline of the future may have some products that are not antibodies. They may be recombinant proteins, but you're not getting to the 10 to 15 grams per liter. That's a big aerial view of the facility in Switzerland. We are actually in commissioning activities. We'll be validating next year, doing the process validation runs. We build 2 cells, but we actually can have 7. We have enough land to build more. Each cell is pretty expensive. So we have to make sure that we know our pipeline products that are coming to continue adding cells to the process. There's a few pictures of the inside of the facility. Telling me that I have 0 time. So one of the things that we did, I'm going to really fast go through is, because of the technologies that we have today, right, all of the closed technologies that we have, closed systems, we are able to put a lot of our manufacturing in CNC space. So 55% of the facility is in CNC space. We've also done supply loops. So we are using a lot of glucose, and so we have, instead of having totes and bags, we have a glucose loop that goes through the facility. We have caustic loops and so on. This is what I was mentioning earlier. We have collaboration rooms. We have analytical labs inside the manufacturing areas. We have 2 central labs, the manufacturing science or MSAT lab and the QC, a high-tech QC lab. It's a facility with a lot of natural light. This is my last slide, so I'm good with the time. So we've also, sustainability is very important for Biogen. So we also implemented a lot of improvements that allow us to have a sustainable features in our Solotron facility. And you can see a little bit of there. And in particular, we have a lot of natural light in the facility with double windows that allow the exterior light to come inside the facility. So that's about about our next generation manufacturing. So I told you we needed to deliver therapies for 1 million patients by 2020, 1/5 of the cost. And how did we do that? We did it by some of the technologies that I described, right? Improving the productivity, eliminating manual steps, having a fully integrated recipe-driven operation, and having that review by exception that I mentioned earlier. So I'm completely out of time, over time, so I'm going to stop here. And this is a work over many years by many people. Somebody asked me that question. We actually got together, all the people that did innovation, say, how do we put all this together to make sure that we can build a facility that can meet our needs? And some of them are in the audience, and I'm looking around and I recognize some of the faces in the audience. So I thank them, and I'm just only telling you what they did. Thank you so much.